So I'm going to do a little bit of a different talk here and talk to you about drug development, but put it in the context of how genomics actually empowers what we're doing at Ceres, but also point out what a lot of folks don't like to talk about, the limitations of genomics and the other work that needs to get done, because in my view, as it has always been, it's not an answer to everything. So first off, a little introduction to the microbiome. Microbiome is a very hot topic right now. There are no shortage of companies forming in the space, lots of popular press in the area. But in the end, when I say the microbiome, I'm talking about the gut microbiome here. There are other entities. And really what we're talking about is that 30 to 50 trillion bacterial cells that essentially live on or in our bodies. And basically, the complex interactions, or really the complex ecosystem that exists by those microbes interacting with each other, as well as with you, the human host. And that happens in a complex array of means, which I will touch on a little bit today. What we're learning over the past 10 or so years, more aggressively, if you will, is that microbes really matter to the health of humans. They play a role in infection control, where basically diverse microbiome allows your body to actually resist infection by various different pathogens, and there's examples of that around C. diff, as well as drug-resistant organisms. They play a major role in modulating inflammation and your immune system. And many different products of microbes actually modulate various different immunological pathways, as well as actually influence the integrity, if you will, of your gut lining, such that it operates functionally. And there's been emerging data that suggests a role of the microbiome in a whole host of inflammatory diseases, as well as some pretty exciting data that's come out over the past year, suggesting that the level of diversity in your gut microbiome can have an impact on how well you respond to checkpoint inhibitors in the immuno-oncology space. And then lastly, metabolic function. They obviously have a huge impact. They drive most of your digestion, and so have a pretty large impact there, and generally regulation of your metabolic system, and as well have been implicated in a whole host of potential health areas there. So in the end, though, really what Ceres is focused on is this concept that basically when those ecologies become dysbiotic, where basically you lose certain bugs or bacteria, you actually have a gain or loss of function that actually then contributes somehow to disease. And so we're interested in developing therapies that essentially restore the microbiome. This is obviously a rapidly emerging both research and therapeutic space. I've already pointed on this. This really accelerated post-2012 when the Human Microbiome Project and the MetaHit projects, which is the European-funded equivalent, published their first papers in this space, which were catalogs of essentially microbes in the human body. At this point, there are literally hundreds of biotechnology companies that claim to be microbiome-based companies, and actually you're now really starting to see the engagement of large pharmaceutical companies as well, although that's still somewhat timid. There's a whole host of different modalities that are being pursued. Diagnostics, obviously natural products made by the microbiome, modulation of the microbiome via small molecules or say prebiotics, or there's also the space where Ceres is working, which is actually the development of actually live biotherapeutics, essentially that are the microbes themselves. So in the end, what are we doing? We're developing what we call ecobiotic drugs. And essentially, in a very simplistic cartoon, you have a healthy microbiome. You have some sort of trigger that leads to a dysbiosis, such that that community of microbes in your gut, to use a technical term, is pretty out of whack. It's not right. And basically, what we can do is we design consortia of microbes that we think can actually shift that dysbiotic state back to a healthy state to essentially have an impact on disease. We have a host of different programs in various different diseases, and we actually are the only clinical stage microbiome therapeutics company out there. So we have a phase 3 trial running in recurrent C. difficile infection. We have a phase 1b study. We just completed a phase 1b study in ulcerative colitis, and I'll talk about these in just a second. We're starting that study later this year, and we have a host of different efforts in other areas as well, including oncology, as I was pointing out earlier, as well as some hepatic stem cell transplant, graft versus host disease. So this is really a novel modality. And I really want to emphasize that because this will come back to to the genomics piece in just a little bit. But what's novel about it too is that you have the ability to potentially modulate multiple pathways. One of the challenges I think with drug development out there is that traditional drug development takes a small molecule and tries to target a particular pathway to have an impact on disease. The majority of diseases are multifactorial and are impacted by a whole host of different pathways. The beauty of a technology of using the microbes themselves as the drug is that these are bugs that have evolved to live in your system and interact with multiple different systems. And so essentially with one drug, you can actually target many different pathways. Here I'm just showing an example of drugs that are used for ulcerative colitis. They target various different pathways, and they also are all subepithelium. Well, microbes and our drugs interact with the gut epithelium and have the ability to potentially signal all of these different pathways. And we're actually developing the data sets to show that that actually is the case. And in addition, it's an interesting modality because it could potentially be used synergistically with existing therapeutics in combination-based type therapies. So, and just to point out that this is not all hocus pocus, this is results from our ulcerative colitis trial. That completed in November, treated 58 subjects. And what you're looking at here essentially is the response rate, where we're measuring clinical remission. And if you look at placebo, subjects given placebo, you can see that you had a 0% clinical remission. If you gave them our drug with a vancomycin pretreatment, we had a 40% clinical remission rate. And we saw dose dependency in that as well. Where if we gave the same treatment on a weekly basis versus a daily basis, you can see a dose response. We have a pretreatment effect here as well, which I'm not going to get into because I don't have the time for that. But basically, this data suggests that actually using a composition of microbes like this in a complex multifactorial disease can actually have a very meaningful clinical input. Just to put this in context, drugs like Humira and Remicade, which are the main drugs for UC, have clinical remission rates around 40%, yet have a lot of side effects. We had 0 side effects in this patient population. So how do we go about designing these drugs? So let me now kind of transition to how we use genomics and the challenges that are out there. So we've been developing a series launched about 6 years ago, and basically we set out to essentially develop a reverse translation platform. And the concept was actually that if you were going to develop a drug in this space and do drug development and take what is very early stage research, the best way to de-risk that process was to actually try to develop drugs by studying humans. Typically, drug discovery starts by in the lab, develop some animal models, test some experiments, get insights into targets, and then to keep developing the drug. And we decided early on that if that was the path we had to take, the company would not be successful. And so we set out to essentially establish a reverse translation pipeline. And what that means is that we actually wanted to do drug discovery de novo in human subjects. And it's really genomics that enables that. And there's very few fields out there that's been the case. That's maybe been cancer is maybe another area where genomics has matured enough and where it's actually being used for de novo discovery in humans. And we're seeing more and more of that. But microbiome was an area where we thought was the opportunity. And so we're basically using genomic-based methodologies to essentially first understand the structure of the microbiome in patients who are both healthy and diseased, and how you change from a person who's healthy to disease or vice versa. Understand the functional properties of the microbiome and the changes in the microbiome, and how, when changes happen in the microbiome, how that impacts the function. And ultimately, then we're designing consortia of microbes that we think can actually restore that function. So there's a bunch of challenges that come along with this. And I'm going to spend the rest of the time just kind of pointing out some of these and showing you some examples. So one challenge is that basically you really need high-resolution genomic methods to basically discover clinically relevant changes in human subjects. And this is a very complex system. And we need to also use these then to ultimately understand drug pharmacokinetics and pharmacodynamics. One, we need to experimentally confirm these causal links. You can use genomics and these other tools in humans to find associations, but to ultimately drive it to causality, you actually need to be able to do follow-up experiments. And so that's a challenge in itself. And lastly, in our case, what we're talking about here is really complex microbiology. Anaerobic bugs that are spore formers. You'll have to take this at face value. People don't know how to grow these things. And manufacturers do not want to manufacture things, especially when they're spore formers. Most manufacturers try to keep anything that forms a spore out of their facility, not generate a facility that makes them. And actually, when Ceres started, really the only places in the world that did manufacturing are places that did anthrax. And so, because anthrax is a spore-forming bacteria. Okay. So let me first talk about challenge number one. So ultimately what we want to do is identify basically and understand the structure of the microbiome as I pointed out. So what we're talking about here is really metrics that measure what microbes are present. And there's a host of different means you can do that, talking about presence of absence of things, total diversity, co-occurrences, phylogenetic structure, et cetera. Next thing we want to do is we understand the function that is associated with that structure. So really these are metrics that measure metabolic and enzymatic activity. And again, you can look at metabolites, you can look at genes, you can look at pathways. And ultimately what we're trying to do at Ceres is explore humans and the microbiome in humans along really two key vectors, that structural vector and that functional vector, and understand how we move someone from a disease state to a healthy state. And so there's a lot of work to be done in terms of how you define those states. What is healthy? What is disease? Basically define what the needed transition is. How far do you have to move someone? And ultimately, when you try to understand this, the methods that you use have a large impact. And so that really brings me to the next slide, is that as we think about it, and many of you who work in the genomics/omics space are probably well familiar with this. Basically, there are all different levels of resolution at which you can work at. If you're just trying to understand who's there in taxonomic, you can do very general 16S-based sequencing where you're going to get information maybe about the genera that are there and maybe the key phylogenetic groups. But if you really want to understand the biology, you need to start moving towards really understanding the species as well as really getting down to strain-level analytics. Because in bacteria, different strains, even of the same species, have different functional properties. In terms of functional resolution, you can do basically gene cataloging using WMS, whole metagenomic sequencing. But you ultimately need to move towards global metabolomics and even targeted metabolomics for the methodologies to really get strong insights. And ultimately what you're getting at here is you go from having sort of you also want to move from really just understanding single organisms to this is a system. We want to understand the ecology. So our analytics, both our data generation efforts and our analytics, need to, at the case of Ceres, operate up here. There's two challenges that come with that. A lot of these are unsolved problems and are really hard. And the second thing is up here costs a lot of money, down here costs a lot less money. Okay? But in the end, you get much higher resolution. So just to give you an example, I'm going to just kind of walk through some examples of kinds of things that Ceres has been working on over the past 6 years that actually make a difference just based on how we generate data. So one area that there's a real need for here to advance this field is improved reference databases. Everybody knows that all genomic methods, and omic methods for that matter, are based on having good reference data sets. Here I'm just showing you an example of identifying the number of species that from our drug that engrafts in a human subject over time. And in this case, we're using metagenomics to make species-based calls. Now, of course, if there are species in our, if we do not have species in our reference data set that might be in the human gut or in our drug, we're never going to detect them in the human subjects. And it turned out when we first set out to do this, we were using the publicly available reference data sets, and we realized we weren't detecting things that we thought we should be detecting. And what we wound up doing then is basically based on our own strain libraries, we generated full genome sequences and rebuilt the reference data sets. And what happens when you do that is you can actually start to detect things that you should be detecting. So prior to rebuilding the reference data sets, you'll see in gray here the number of species from our drug that we detected in subjects over time. And once we revamped our reference data sets, which was not a trivial undertaking, and you can see the blue represents the species that we detected. And so basically by improving our reference data sets, we were able to basically get much higher resolution in our various analytics. The second thing really is that ability to work at that higher level resolution. So here I'm using an example of strains. So basically, existing methods for essentially identifying bacterial strains in a complex composition are a science unto itself, and it's a big challenge. We've been working on strain-based analytics at Ceres, and basically, this is allowing us to get higher resolution insights into the changes in the microbiome. And just at a very cursory level, I'll show you here. Here you're looking at a bunch of different subjects this is from our ulcerative colitis trial. And if you just used a species level analytics to try to detect who's there in terms of the bugs, that's represented by these blue bars. If you actually then use strain-based analytics, where you establish source-sink relationships between what's coming from your drug versus into a subject, you can actually start to identify additional species that are in the signal. So again, you got to keep kind of drilling down and work at a pretty high level of resolution to basically be able to get the insights that you need. The other thing you need to be able to do as you think about developing a drug is it's not just about characterizing the system. So in an academic setting, typically what you're doing is you're seeing a lot of papers come out where people are describing the microbiome, describing these changes, looking at these functional differences. But in the end, if you want to design a drug, you have to decide what are the critical microbes that need to be there that will basically allow you to get the functional response that you're looking for. And so what we've been developing at Ceres is a whole host of algorithms which are really designed to try to detect the simplest set of microbes that could lead to a specific functional response. And as we've built these algorithms and started to look at these data sets across our subjects as well as public data sets, you start to find some pretty interesting patterns. So one thing is when you look at microbiome modulated metabolites, you actually see that there are certain metabolites that basically have a large number of sorry, yes. So you have for basically, sorry, the axes on this were flipped from what I thought they were. So basically, what you're looking at here is basically you find certain metabolites for which only a couple species or single species have the ability to generate. So in this case, there's a single species that there's about 40 or so metabolites that can only be made by single species. There are other metabolites that are much more broadly distributed across the various different bacterial species. And so, for example, here, you've got this set of metabolites that's represented here basically can be made by 25 or so different species. And so what we're starting to do is dissect out that understanding of when we want to get a certain functional response, how complex does our drug need to be in terms of the diversity of bugs that are in it to get that functional response? And you can see that same kind of pattern, a slightly different distribution, when you actually start to look at that just based on gene content. Again, there are certain genes that are represented in a very, very small number of species, and other things that are very broadly distributed across that population. And so those understandings have relevance for how you actually design your drug. So challenge number 2 is really that need to experimentally confirm the causal link. Okay? And I really want to emphasize this. This is where I want to kind of stress it's not just about the genomic readouts. In the human subjects, we can find associations, we can have understandings of how different bugs relate to different functional properties. But in the end, you got to really demonstrate and go beyond that in the lab that basically what your hypotheses are correct. Right? And what that takes doing is first just having the bacteria to work with. And in the microbiome space, this was not a trivial undertaking. Many of the bugs that were identified through the human microbiome genomic sequencing efforts that I talked about earlier did not exist as cultured organisms. So genomically, we knew a lot of things were there, but we had no isolates of these things in our libraries. And so that's obviously a huge limitation. Both not just on what you can do experimentally, but also on the reference data sets, as I was talking about previously. So what Ceres set out to do was to build a comprehensive strain library of the human gut. We now have a library that constitutes about 35,000 isolates, which represent over 450 species. Just to put this in context, I'll point out 2 points. One, 12% of our isolates are novel species that have never been previously cultured or even known to exist. We have no references of them anywhere in any public domain. So we're learning about totally new biology. Basically, if you look at the Human Microbiome Project, they published what was called the Most Wanted list. That was a list of bacteria that they knew were in the human gut based on genomic measures, but no one had a culture of that they could generate a full genome sequence of. Ceres now has in its library, that represented 485 species. We have over 60% of those species in our library. Okay, so again, we have the ability to do novel experimentation. Just to put this in context, this is a 16S-based phylogeny of all bacteria known to exist in the human gut or be associated with the human gut in some way. The red ticks here around the outside essentially represent basically species, sorry, wrong one. Yeah, red ticks. The red ticks around the outside here represent species that we have in our library. So you can see we have a lot of the diversity of the human gut in our strain library. And actually, as I said before, strains matter. And these orange ticks around the outside represent all the species for which we actually have strain diversity in our library. So we don't just have one isolate of that species, but we have many, many different. And in the end, what we spend a lot of time at Ceres doing then is actually functionally characterizing all these different strains. And I'm not going to get into the details of the many, many different ways that we do this as delineated down below here in terms of using genomic methods, measuring adaptive immunity, inflammation, et cetera. But what I want to point out here, this is an example of bile acids, is that when you go and look at different strains, where each row here is a different strain, from a single species, and you look at different bile acid metabolism properties, for example, you start to learn that blue means it can do it. Different strains have different profiles in terms of their ability to metabolize bile acids. But in addition, some have pro-inflammatory responses and others don't. So for example, this set of strains here actually also has some pro-inflammatory, so it'll create an inflammation response in human subjects, whereas these strains up here that are noted in green don't. And so if you're selecting a certain strain for a drug that you want to metabolize bile acids, you're going to select the one that hits the bile acids that you want, but also doesn't have a pro-inflammatory response. And so what we're doing is building the kinds of data sets that allow us to actually bring both genomic data from human subjects in combination with functional data such as this to basically be able to then predict and design drugs. And as well, the other challenge we have, obviously, is that basically we need to develop the systems, the experimental systems to do this in. And for the sake of time, I'm not going to get into this, less just to say that we're developing novel ex vivo systems to study epithelial cell biology, and we're also developing new animal models which allow us to actually have a human microbiome established in the mice to then run experiments. Because obviously, if you try to run an experiment with a mouse microbiome, it's going to be a little hard to understand how human microbes are behaving properly. The last challenge that I'll end on really, again, is that ability to make, and I touched on this before already, it's the manufacturing challenge. Ceres is actually built in-house a full good manufacturing practices facility, basically, so it's regulatory, it meets regulatory snuff. I'm not going to go into the details here, less to say that we're dealing with a lot of complex biology, and we've been developing the methodologies that are needed for drug release, for analyzing the purity, potency, and pharmacological properties. So I'm just going to wrap up really by kind of summarizing what I think are some of the key challenges and gaps, as well as really, in the sake of this meeting, opportunities for academic industry collaboration. These are really hard problems. All of these things I'm talking about are not going to be solved by one company. We're doing a great job, I feel, at Ceres in making a dent in many of these things and moving some drugs forward. But these are really challenging problems that are going to require a lot of different people working together. So one, obviously, is improving the metrics that we have to get more meaningful insights into the microbial ecology. And particularly there's a real need for investment in reference and control data sets, and as well developing methods that are cost efficient. We need to establish benchmarks for clinically relevant biological changes in the microbiome. Again, it's not just about measuring something, but it's about actually then validating those measurements. And so there's really a need for in silico methods to identify these changes and analyze these complex data sets, but as well as the experimental systems. And then lastly, I'll point out here really is that manufacturing piece. So yeah, so with that, I'll end, leave it there. So
thank you.
Thank you.
So, have you found any fungi in your gut
Yeah,
microbiome?
it's classic. Yep, get asked that all the time. So obviously the microbiome is a complex system that involves both bacteria, viruses, and fungi. As a company, we've been focused on the bacterial component because, again, being a small startup company, so to speak, well, I guess we're not. We're a little beyond that. We're about 150 people now and relatively reasonably funded. So, but nonetheless, there's still only so much you can do, right? So we've been focused on the bacterial component, but certainly are generating the data sets that would allow us to look at the fungal component as needed.
It's a system, right? That's the first premise of the whole.
Absolutely. No, absolutely. Yeah. In the diseases that we've been focused on, fungi are not known to play a significant role. There are some diseases that I can consider us engaging in where that's not necessarily the case.
To that point, do you see any endofungal bacteria?
Yeah. So again, yes, they can be there, right? Absolutely, but not something that we've been focused on as a company. So I don't have too much to say about that. Being someone who used to study fungi as a grad student and looking at essentially endophytes, so tree-associated, fungi, certainly an area of particular interest though for the future. Yeah.
It's very interesting. And so, in an effort to characterize the effect of micro strain and discover the disease-causing microbes. And since it's dynamic, the microbiome system is dynamic, complex, how do you determine the effects of the interaction of multiple microbes and just individual microbes, right? Together, consistent. Also, the reference genome will be dynamic too. And when you eat a bite in different diets or in different time, might affect the environment. So how you go about
Sure. Great question. So you're absolutely right that the microbiome is not static. It's a system that has variability in it and variability through time. So one of the first things that we focus on in the company is what I like to think of as interventional data sets. Many of these large cohort studies are just study a bunch of people at a snapshot in time that are healthy versus diseased. We're much less interested in those data sets, not to say that we don't use them and that they're very valuable, but we're interested in these interventional data sets. So in other words, time series. How does the system move from a state of health to a state of disease? How does someone move from a disease state to health? If someone goes on an antibiotic treatment, what happens when they go on that antibiotic treatment? If someone goes on an anti-inflammatory or comes off an anti-inflammatory, what happens? And so we really try to look at, and our tools are mostly geared towards thinking about essentially time series and variation in the context of that. And then, the bugs themselves are living organisms. Right, that adapt. But we spend a fair amount of time really trying to understand, like I said, that strain diversity and how that varies both across strains and how that also might vary based on different culturing conditions, et cetera. And we're building that kind of knowledge into our drug design algorithms that ultimately define compositions.
Also, the system often reverse back to the regional, some region, for example, some people actually took the drug. And
sure.
They're healthy, and then go back after a while.
Yep.
So how do you go?
Absolutely. So ecological systems absolutely have resiliency in them. That's what's typically referred. Most of the research that's been done, and I would say Sirius has found this ourselves in our own clinical studies on subjects who are on placebo, that resiliency is only goes so far, right? So while the system may rebound to some extent, it typically doesn't get all the way back to its original state. And I think it's an evolving science. I mean, there's still much to be learned. But I will emphasize there, actually, on that point, is that we're starting to see work come out where people are publishing clinical studies where they're sort of using, let's say, FMT, fecal microbial transplants. The issue with those studies that I have is they're not doing placebo controls, right? And basically, for just the point that you're pointing out and others, lack of placebo-controlled studies is a real issue. Because I think the insights that you draw from the conclusions without that placebo control and knowing what that natural variation may be is suspect.